This video demonstrates a procedure to isolate high-purity viral nucleocapsid DNA from infected cells. First, lipid membranes are denatured using a dual Freon extraction. The sample is then ultracentrifuged to pellet the viral nucleocapsids and separate them from lighter cellular and viral components. Detergents and proteinase K are added to release the viral DNA from the pelleted nucleocapsids. Then dual phenylchloroform extractions are performed to remove all remaining proteins. Finally, the viral DNA is precipitated in solution. Ultimately, the results show the highly concentrated and pure viral DNA has been isolated from these infected cells.
The main advantage of this technique over existing methods that do not involve Freon extractions or ultracentrifugation is that the viral DNA produced here has very low levels of host DNA contamination, making it ideal for downstream procedures such as PCR and deep sequencing of viral genomes.
We first used this method in the early days of recombinant DNA work when extra precautionary measures were of high importance. We developed this procedure to isolate viral DNA while working in a glove box under BSL-4 conditions.
Visual demonstration of this method is critical because the gradient formation and DNA ghosting steps are difficult to learn. This is because layering the gradient requires an extremely steady hand, and the DNA ghost that precipitates in solution can be hard to see if you don't have prior experience spotting it.
Begin this procedure by preparing 5 to 10 15-centimeter diameter dishes of tissue culture cells for infection. Here, PK15 cells are prepared for infection with pseudorabies virus. When the cells are 95 to 100% confluent, replace the medium with 4 milliliters of medium containing virus at an MOI of 5 to 10. Incubate